Bismillahirrahmanirrahim. Welcome again, guys. Welcome to another video lecture by Biology. Today in this video lecture we are going to talk about types of restriction enzyme on the basis of their source of isolation or their biochemical activity. In the previous video lecture, if you guys remember, we already talked about the restriction modification system. We talked restriction enzyme properties, and we also talked the introduction to restriction enzyme. If you miss these videos lecture, it is present into my channel. You can go back and watch it. Don't worry, I will give the link in the description box, then you will check it. Okay, so let me clean the whiteboard and let's start. Okay, so let me write types of restriction enzyme on the basis of their source of isolation or biochemical activity. So let me write source of isolation or their biochemical activity. Okay, so according to that we have almost three types of the restriction enzyme. So let me write each name. The first we call that isoschizomer. Second we have Neoschizomers, and third we have Isocaudomers. Now these are the three main types of the restriction enzyme according to their source of isolation and their biochemical activity. So let's explain the first types, which we call that isoschizomer. Now as the name indicate, isoschizomer, in this case the source of enzyme means SOURCE, source of enzyme is different. So remember, the source of enzymes, usually we have the pair of enzyme which we isolate from different bacterial cell, but their recognition is same. Recognition and the digestion of the DNA is same, while recognition and cleavage or digestion is same is called isoschizomer. As the name indicate, iso means same, so they have same recognition site. Two different enzyme which you isolate from two bacterial source, but they have same restriction site as well as they have same recognition site. For example, we have a DNA molecule, let's say in this case. So let's say these black region can represent the recognition sequences. RS, recognition sequences, or okay. So we have two source of restriction enzyme. So let me draw here. It's the first source and this is the second source. Source number 1 and source number 2. These are actually restriction enzymes. R1, R2. So they will recognize the DNA molecule. This is the DNA molecule. Let me write. Okay. Now these restriction enzymes have same recognition site as well as digestion. So let's suppose both have same recognition, as you can see. So they will recognize that specific area. And when they digest the same area where they recognize, let's suppose they recognize that specific area, and when they make a cut or make a cleavage at that specific point, now these source of enzyme are called as isoschizomer. So they will make a cut at that specific point. Let's say it make a cut, as you can see. Okay, and after that what we will get, we get two different or two fragment of the DNA molecule. Okay, this is called isoschizomer. In this case we have the example of so we have the example S, P, H, L and B, A, U, L. Okay, now these are the restriction enzyme which is isolated from different source of the cell, and they have same restriction site as well as recognition site. So they are isoschizomer to each other. Okay. Next we have Neoschizomers. Neoschizomers is the type of restriction enzyme which have the same restriction site but different cleavage point. Let's suppose again in this case we have the DNA molecule. Okay, now let's say these are the recognition sequences or S, recognition sequences, and these are the restriction enzyme which is isolated from two different sources. Means this is restriction enzyme 1 and this will be restriction enzyme 2. So it will be isolated from two different sources, source 1 and source 2. So they will recognize the same restriction site. Both restriction enzyme, when they, means we have pair of restriction enzyme, when they will recognize the same recognition sequences as recognized by the restriction enzyme 1, and both restriction enzyme have different cleavage point. Suppose this one restriction enzyme will cleave that specific area and this one restriction enzyme will cleave that specific area. So this is restriction enzyme 1 cleavage point and this will be the cleavage point of restriction enzyme 2, but the recognition site or the recognition sequences will have the same, but the restriction site will be different. We call that a Neoschizomer. Okay, now example of this we have the example S, M, A, L and X, M, A, L. These are the Neoschizomer to each other. Okay, third we have the Isocaudomers. So in this case again we have two source of the restriction enzyme which is isolated from different bacterial cell, and when they have the same cleavage point, remember when they have the same cleavage point, try to understand the concept, but they have different recognition site. Suppose again in this case we have the DNA molecule. Okay, now let's say these are the recognition sequences, as you can see. This is recognition sequences 1 and this is recognition sequences 2. Okay, and we have again two sources of the restriction enzyme, as you can see. This will be restriction enzyme 1 and this will be restriction enzyme 2. And let's suppose they have the cleavage site. So let's say they have the cleavage site at that specific position. This will be the cleavage point or digestion. Okay, so this restriction enzyme 1 will recognize that specific area and this restriction enzyme 2 will recognize that specific area. When two or many restriction enzyme will recognize different restriction site in a DNA molecule, mean different recognition site in a DNA molecule, but they can cleave the specific area. Both can cleave the same area, as you can see. Then this one enzymes are called as isocaudomers. Okay, now the example of, in this case we usually get the blunt end as well as the sticky end. Okay, so in this case the DNA will be produced in this form. Let me write. So this will be the blunt end, as you can see, and the DNA is also produced in this form. Okay, so this is the sticky end, as you can see. So let me write. Remember, in this case, they will produce the blunt end of the DNA. So when we use isoschizomer, so they will usually produce the blunt end of the DNA molecule, blunt end. When we use the Neoschizomers, so they will produce the sticky end. Sticky end. And when we use the Isocaudomer, then they will produce sticky end as well as blunt end of the DNA molecule. Okay, the example of this DNA, the example of Isocaudomer is BAM H1 that have the cleavage site R. So the complementary will be CCTAGG. So in this case, they usually make a cut sometime at that specific point, or sometime they will make a cut like in this way. So it means that sometime they will produce blunt end and sometime they will produce sticky end. There is another example. We have BC2. So the restriction site will be GATCA. Their complementary is ACTAGT. Okay, so they have the same as we explained in the BAM H1. So they will also produce the blunt end as well as the sticky end, as you can see. Okay, now that's all about the types of restriction enzyme and their example as well as their properties as well. So I hope you guys get idea about the types of the restriction enzyme: Isocaudomer, Neoschizomer, isoschizomer. But if still you have any kind of confusion and question, you can write it into a comment. Inshallah, in the next video lecture, we are going to talk about some other types of the enzymes, some other types of restriction enzyme, which we call it type 1, type 2, type 3 and type 4. So we will be talk it in our next video lecture. So please stay tuned and continue to watch the video. Thank you.